Welcome all. It gives me immense pleasure to introduce Josef Coresh to you. Joe Coresh is the George W. Comstock Professor in the Department of Epidemiology at the Johns Hopkins Bloomberg School of Public Health. He directs the Cardiovascular Epidemiology Training Program and the George W. Comstock Center for Public Health Research and Prevention. He's a core member of the Welch Center, which many of you may have heard about, for prevention, epidemiology, and public health. He has joint appointments with the Departments of Medicine and Biostatistics. When he was named the inaugural George W. Comstock Professor, the appointment not only had a personal meaning for him, it meant he would be able to invest in innovative research that lacked conventional funding. Following in the footsteps of his mentor, Coresh is now making great advances in research about dementia and kidney disease, as well as other vascular diseases. So he obtained his MD and PhD degree in epidemiology and his master's in biostatistics at our university in 1992. And following his postdoctoral training, mentored by Dr. Clagg and Welton, he joined the faculty and was promoted to professor in 2004. He has many contributions in the national arena, but I would like to mention some of them. He served as the vice chair of the influential 2002 National Kidney Foundation Practice Guideline on the definition and staging of kidney disease. He worked closely with other experts in leading the Chronic Kidney Disease Epidemiology Consortium, which published the definitive equations for estimating risk. Listen to this. He leads the CKD Prognosis Consortium, which includes over 50 cohorts and 3 million participants globally. CKD Prognosis Consortium results inform global guidelines on CKD staging and updating the definition of CKD progression outcomes in clinical trials. He has led the Hopkins component of several key national and international studies, the ERIC Neurocognitive Study since 2010, and the CKD Biomarkers Consortium since 2009. Needless to mention, he's recognized as an international expert in kidney and cardiovascular disease epidemiology with over 400 research articles which have been cited over 50,000 times, and is recognized by Thomson Reuters as a highly cited researcher in clinical medicine among the top 1%. He's received multiple awards from the American Heart Association, the National Kidney Foundation, including the AHA Lifestyle Council Mentorship Award, another duty that he takes really deeply to heart, and the NKF 2014 David Hume Memorial Award. With that introduction, we have the great honor of listening to you.
Well, thank you very much for the introduction and the invitation. It's a pleasure to speak to you, and I guess Hopkins has been good to me, so we'll figure out maybe why. And I've tried to sort of figure out what I could impart by way of lessons from the past 30 years or so. So I came to Hopkins, I just lectured to the medical students for 2 hours, and I came and sat in those seats in 1986, right? So it's been a little bit of time, and it's been quite lucky. I've met wonderful people, and it's really become sort of my academic home, but also my home and kind of a second family. So I thought I would talk about a few topics. Why epidemiology and vascular disease? Why did I choose that? Why me? I got invited, and I was thinking, so why am I being invited, and what have I done? So in some sense, that's central to what I'll talk about. And the biggest contributions have been in chronic kidney disease. So I'll talk about chronic kidney disease. And we were lucky to address fundamental questions and gaps. And maybe the lesson is, why are these gaps? What makes gaps? How do you actually answer such questions? And why were we lucky? And then I wanted to talk briefly about what I actually spend most time on, which is the ERIC cohort, the ERIC Neurocognitive Study, and our new ACHIEVE clinical trial, which sort of tells you a little bit more about this voyage of research, which I think is not fully predictable, but has been quite enjoyable. And then finally, some summary slide and maybe some thoughts about the future. And you know the famous quote, predictions are particularly hard, and especially hard and risky about the future, so with a word of caution. So that's sort of the general outline. So why epidemiology? So what is epidemiology, right? I suspect most people in this crowd are not epidemiologists. I actually came to Hopkins not knowing what epidemiology was. I was somewhat exposed to issues of public health, but it wasn't until I took Leon Gordis's course that I was like, oh, this makes sense. It's totally obvious. And then I realized, well, if it's totally obvious to me, maybe that's what I should be doing because it kind of makes sense to my mathematical background. So epidemiology is the study of the distribution and determinants of health-related states or events, including disease, and the application of this study to the control of disease and other problems, right? And it's sort of this opportunity to do control and prevention that was really exciting. It's central to public health, evidence-based medicine, disease prevention, risk factors, study design, analysis and interpretation, and causal thinking in a complex human situation. So for me, that was attractive, and for me, a lot of the fact that often when I was doing my early rotations on the wards, a lot of patients are very sick. And for some things, we can do amazing things and work what seem like miracles. And for some chronic diseases like heart disease and kidney disease, once a lot of the damage is done, you realize that really, if you could have prevented the disease in the first place, you may have a lot more room. So that was attractive to me. And really, we need to work throughout the spectrum, but I'll give you some lessons about sort of where we've done our work. So second, why cardiovascular epidemiology? What drew me to this field? And basically, cardiovascular disease is the leading cause of death. So it seemed like at least I would know I'm working on something important. But people were doing well. So this is this graph. If you look at historical trends for age-adjusted death rates for major cardiovascular disease, really the epidemic rise of coronary heart disease peaked in the late 60s. And since then, we've had a dramatic decline in coronary heart disease. It's interesting that stroke rates have been declining for the whole century. And then deaths from hypertension with treatment have really declined very, very markedly, right? So we're doing well, but in some sense, to me, that was exciting because we were doing well, there were good clues, but it was still the leading cause of death. So there must be a lot more gain to be had. So for me, a lot of the lesson is really how do you continue to innovate when there's some progress already there? Even though it's very interesting talking to some other colleagues who've taken on some causes in cancer, some people have been drawn to the opposite where they say if it seems hopeless, that's where people are needed to create hope. So I think there's ways to address different kinds of problems. So then next, when I was preparing for this talk, I was sort of, why me and what should I talk about, right? So in some sense, it gets to sort of what are answers that are useful, what is impact, why do people care? You know, you work every day, and every day you just do a little bit. George Comstock says, you know, win small victories one at a time and try and do something for humanity. So I centered this on impact. This statistic has sort of been kind to me. Citations to co-authored papers are a measure of impact. It means somebody read your paper and decided to cite it and use it for something else. And I'll talk about why I think some of these things have been cited. So I downloaded this from Scopus, and basically this is a graph with the papers on the x-axis from 1 to about 500 here, which is a whole other thing. I've never thought I would be involved in this many investigations, but thank God it's really co-authored papers. I did not write all these papers. People I worked with wrote most of these papers, right? And so some of these papers are clearly having much more impact. On the arithmetic scale, the biggest one is the CKD staging guidelines. And that guideline, chaired by Andrew Levy, with me as co-chair in 2002, has been cited now about 9,000 times. And then the summary of that has been cited about 3,000 times. And then an explanation of that has been cited another 1,000 times. So clearly, this particular staging guideline was fundamental. So guidelines, my lesson is, are important. And then there are certain ones where there was a pivotal point, I'll try and show you why, where it seemed like people really paid attention. Then we made a paper in 2003 on the chronic kidney disease prevalence, right? And establishing the prevalence of disease in the population is a very fundamental, very simple epidemiologic quantity. So I started my career doing genetic epidemiology, very sophisticated models. I'll show you that. That's one of the earlier papers. And I was very fascinated by all of that. When I started doing this, I realized there was sort of this big E epidemiology, the big picture, very simple statistics, right? Or at least simplified to a bottom line message. And often the impact has been greater when the message is about something that people really care about, but the answer is fundamentally understandable in a relatively simple way. Then we worked on calibration of estimated GFR. It turned out that estimated GFR was central to the staging system, and getting it right was important, so methodological papers were important. Then we worked on trends in the prevalence of the disease in the US, and that was again very highly cited. Then we decided to try and see if the old equation for estimating GFR was correct, and I'll show you that work. And it turns out that has become the definitive equation for estimated GFR globally. So that has rapidly, published in 2009, been highly cited. Then we established the CKD Prognosis Consortium, and I'll tell you about that. And the first paper in green by a trainee, Kuni Matsushita, was a fellow when we started that, has now been cited over 1,000 papers. And working with bright fellows has really been central to a lot of this, and it's kind of interesting, and that's what makes Hopkins special. And then now we've been working on trying to help the FDA in the treatment of kidney disease, and that's a paper we published in JAMA in 2014, and you can see it takes time for things to have an impact, and citations may not be a very good measure. They're only correlated with impact. Some things could be more fundamental in a different way. In between, there were other guidelines that we've worked on that expanded on the work on kidney disease. And then on the bottom, a sort of work on genetics and omics that I've been interested in on and off for the past few centuries. So looking at this list, it was interesting that my two papers for my doctoral dissertation, right, it turns out the story of the first papers, I was interested in genetic epidemiology. I thought it would be promising. It seemed clear that there was variation in risk for heart disease. And apolipoprotein B was the major protein on LDL, the major particle for causing atherosclerosis. So I worked with Stelios Antonarakis, Peter Kwiatovich, and Terry Beatty to figure out the genetic basis of elevated levels of ApoB, which was thought to be the major risk factors. We did a segregation analysis, which at that point was thought to be the definitive way to look at genes. We did a linkage analysis, which was thought to be the most valid. I submitted both of those papers to American Journal of Human Genetics, and they said, oh, pretty nice, merge them, make them into one paper. I was like crushed. I thought I worked for 3 years, 2 papers, they're going to make them one, and each one was long. And Stelios said, well, they're going to take your paper. This is good news. You should accept it, right? And that may be one of the lessons. If they accept it, just listen and do what they say. So we merged it into one paper. The interesting lesson to me is when I looked at it yesterday, it turns out that paper has only been cited 15 times. The paper next to it was just the epidemiology of ApoB and triglycerides and which one is more likely to be causative, and that's the one next to that, which has been cited a couple hundred times. So that's been kind of nice. And that question has actually been difficult. For me, I actually then realized some of the weaknesses in what we were doing with the genetic epidemiology, and even though we had gotten a big grant with my mentors to continue that work, I believed that the fundamental assumptions we were making were not ones that I could deliver on. So then I ended up meeting with Michael Clagg and Paul Welton. I had done some side papers with them, and they said 2 important things. Paul Welton said, kidney disease is an epidemic, and hardly any epidemiologists are working on it. And that seemed to have been sort of the key to deciding to move from lipids to kidney disease and think about that hard. And it happened to be lucky because really there were few people working on it fundamentally. It did mean that the early papers were completely rejected. So, and then Mike Clagg said, you should work with good people. And I thought it was self-serving because at that point, Mike Clagg was an amazing person, but he was an assistant professor. His biggest study was the precursor study, which had 1,000 people, which I thought was too small, and his grants were small, whereas the genetic people had a lot more money. Turns out I liked him, I worked with him, and I think he's right. If you can work with really good people, good things happen. And in hindsight, he went from assistant professor to vice chair of medicine to the dean of the School of Public Health, and his career progressed, my career progressed, and we've sort of worked in collaborative ways all along. So you never know what gambles you take. In the way of these other genetic papers that I won't get to talk much about, was another gamble, having been disillusioned with linkage analysis. When there was a call for papers to look for genes for kidney disease, I went to Mike Clagg's office and said, we built a specimen bank. I don't have time to do this. And I heard about this technique called MALD, mapping by admixture linkage disequilibrium, which would do an association test, which was initially viewed very disfavorably. But I thought for kidney disease, where the risk gradient was that African Americans had much higher risk than European Americans, and for hypertensive kidney disease even higher, and we had tried to explain it with epidemiology and we couldn't, I thought there could be the rare chance that some variant, kind of like sickle cell disease, would be at a higher frequency in Africa, would explain that, and would explain a genetic component of kidney disease, was an interesting strategy. And I thought nobody else was going to go in with that strategy. Everybody else would go in with family studies, linkage analysis, and it turned out everybody else went in with diabetic kidney disease, which was sort of the most important thing, but it turned out was incredibly hard to crack. So we tried that. It turns out they wanted to kick us out of the consortium because they thought the method was crazy. They protected us. They didn't kick us out. Linda Cao had come back from time in Taiwan and spent the next 6 years with Mike Clag, Roland Perec, and others really pursuing that studies. People at Harvard perfected some of the techniques. People developed the right laboratory methods for that. And 2 papers simultaneously in 2008 published the same result, that one locus was going to be this locus with an excess allele frequency for kidney disease risk. It turns out then we looked in that locus, and smarter people looked next door and found out that that was APOL1, not MYH9. And then it turns out that is a high frequency allele with a high relative risk for a common disease, which turned out was the thing I thought was the exception for heart disease and the reason I sort of didn't stay in that field. So they got lucky and did a wonderful thing with that, even though that's very fundamental, but it's sort of this peak here, which is not as tall as some of the much simpler things that we've done. Later on in 2007, I was doing epidemiologic studies working on kidney disease, and genome-wide association analysis came around. And it became clear to me that with 1 million markers, you could do a lot more than what we could do in the early studies with 3 markers. That technology was really a lot of other people developing all the fundamentals of that, but as people in public health, we understood the cohorts, we had the samples, and I was interested in kidney disease. So Anna Kotkin, who was a fellow, worked with me and Linda Cao, learned the techniques, collaborated with Carolyn Fox, and then published a number of genome-wide association studies. And at this point, she heads the global consortium with some partners for the genetics of chronic kidney disease. And so they published that paper, and this is sort of the peak next to this other tall peak. And then another fellow here, Georg Ehret, worked with Arvind Chakravarti on GWAS of blood pressure, and that became highly cited. Anna then did follow-up work on genetics of kidney disease. And then later, it turned out we were doing the ARIC study, and it turned out NIH did not want to renew visits for the ARIC study for heart disease. Right? So they had done visits for a decade, and then they decided, oh, well, it's more exciting to start another study, so let's reduce the money and just do follow-up. We became convinced that it was really important to re-examine this aging cohort. So then the question was, what's important enough to justify the investment, right? And in some sense, because it was funded by NHLBI, tackling the heart, which was important, was going to be hard because they get to decide on the heart. But Richie Sherritt had looked at vascular disease across different beds. We were interested in kidney disease. He was very interested in brain disease and microvascular disease in the brain. And it became clear that there was a growing literature that clearly dementia is an issue. Right? There was the discovery, I remember going to the lecture where ApoE was discovered as a risk factor for dementia. So that was the whole explanation for Alzheimer's dementia. But clearly the vascular contribution through big strokes and more importantly, many small stroke to cognitive decline was increasingly quantified. It was also clear that if you measured the blood pressure at old age, you didn't see association. But it was less clear of if you measured the blood pressure 20 to 30 years earlier, would you see the risk factors for late cognitive decline? And importantly, if that was true, it would open a door for prevention of cognitive decline. It would also mean that it was the kind of door that couldn't easily be done in clinical trials, whose average duration is about 3 to 5 years with longer follow-up a little more. But if really kidney disease and brain disease like it was a multiple decade process from midlife to older age of deterioration, and to do prevention you needed to start in midlife at age 55 to prevent events at age 75 plus, that would be societally important, powerful, and conveniently exactly the study we had, right? Because we had the measurements in midlife, then we had this big gap, and now we had a cohort of older people. So we rejuvenated that, submitted a large grant, and we're fortunate to be ahead of a big wave of investigations doing some very good work in this area, and it's pretty clear that that hypothesis is true. And Rebecca Gottesman now co-leads that study. She's in the Department of Neurology and knows a lot more than I do about dementia, and that's kind of some of these papers later on. So there's sort of a theme of collaborating and looking for opportunities and being fortunate at Hopkins to work with multiple people. One of these other papers are trends in diabetes, where we took the kidney disease trends and translated that to diabetes. And the major studies I work on are the CKD Prognosis Consortium, the ARIC Neurocognitive Study, and then I've recently gotten involved in the Global Burden of Disease project, where my contribution is minuscule, but their papers are highly influential. And then this is sort of looking at the same data on the log scale. It is funny how numbers sort of have different appearances when you look at them in different ways. You look at this and it looks like I've been doing nothing for all the time in between. It turns out actually you work steady day to day, and it turns out early on there are a lot fewer papers, and then over time there are more papers because the author groups get bigger, and then every once in a while things are sort of very big impact papers and lots of citations, over 1,000. But really, it's just the tip of lots of work day to day, trying to make a difference, trying to be clever on different things. So why did our work have an impact, right? Trying to summarize in one slide. We got a presentation recently that says, you know, rather than saving it all to the end, just tell people early and that's it. So if you want to leave after this slide, that's fine. I think it was sort of opportunity, which has to do a lot with luck. Timing, in that we were early and very little was done in kidney disease. And really the most is collaboration. So I'll talk a fair bit about collaboration in these large groups. And I think the challenge is how to collaborate effectively, right? And when I heard from some colleagues about the fact that often epidemiology is team science, my first thinking about team science was, that sounds kind of boring. It's a bunch of anonymous, you know, nothing happening. But really over time, I've come to realize that it really is that. You often build on what other people have done before, using tools that have been developed before. You get to do some innovative things, move the field ahead, build to the next thing. But I think the key is that the teams are like professional sports, right? Where to make something happen, where you've got a few people on the field, they may be scoring points, but then you've got people directing them, and they have to be very good. Then you have to be people managing the business, and they have to be very good. Then you have to have people who manage the stadium, and if there's no stadium, then people can't attend, and they have to be very good. Then you have to have people who are cleaning the stadium. If that doesn't happen, they have to be very good. Then you have the TV, and they're very good. So actually, when you build a team, really all the components are incredibly valuable, right? So I was just recently with our staff in Hagerstown, and I really believe it. Without them, we would not have the data. And without valid data, we can't do anything. So at least in epidemiology, the teams tend to be large, and the papers are driven by certain key people, but it really takes a whole team of excellence to get things to move forward. I think then in addition to that, I think we've had some numerical creativity, and hard work sort of never hurts. It was helpful that ESRD, end-stage kidney disease, as an epidemic was recognized and costly, but its underlying denominator, the chronic kidney disease in the population, was poorly defined, so it was an opportunity for an epidemiologist. And broad collaboration can lead to results that address an important question, provide robust, useful answers, and stand the test of time. So I think that's kind of what I'm looking for these days, is good questions. And I've now come to learn that actually the best questions are ones that other people tell me. And then, you know, hopefully the challenge is to figure out whether the question is answerable. And then when you get an answer, to get an answer that matters and stands the test of time. So in terms of the collaborations, really I've been fortunate that Mike Clegg and Paul Welton were sort of founding the Welch Center, which stands between the School of Public Health and the School of Medicine here at Hopkins. So being at that interface has been tremendously fun and a good opportunity. And then there, I've worked with a lot of cardiovascular epidemiologists in our group, pictured on the left, and then staff at the Comstock Center who collect our data. And then there was sort of an amazingly fortuitous collaboration and meeting with Andrew Levy and Leslie Inker at Tufts, who really provided the key insights and link to influence in nephrology, where they needed my insights that were epidemiologic and numerical, right? And so that became a good synergy of the two. And it really is useful when you have partners that are both respectful and very different from what you do. So back to the science of this. A few examples of the kinds of things we've done, the kinds of questions we've answered, right? So when I entered the field, it was clear that end-stage renal disease was an epidemic, that a lot of emphasis was done on the numerator. End-stage renal disease prevalence was increasing. By 2010, there were 600,000 patients on dialysis or transplantation, and there was an assigned cause by the physician of diabetes or hypertension. If you take into account the denominator of the whole U.S. population, then it becomes clear that there's a difference between looking at numerator, where you see that the most cases are white, the next most cases are black or African Americans, but the rates among African Americans are much higher than the rates among whites, right? So this was something to be addressed. How much of this was socioeconomic disadvantages, disparities, hypertension, diabetes, and then the potential for residual genes, where I told you we worked on later, right? It's also true that I entered somewhere here in the 90s where the epidemic was scary to all of us, and it turns out by the time my work was more maturing, the epidemic was really flattening out. But by then, we were turning to the denominator of the whole population of kidney disease that may be driving this. So then I'll talk about sort of three fundamental questions that we got to address. How common is kidney disease? What happens if kidney disease isn't treated? And how do we improve treatments for patients with kidney disease through faster clinical trials? So how common is kidney disease depends on what the kidneys do. So the kidneys filter blood. They get about 20% of cardiac output, right? So for a vascular epidemiologist, even though the heart is the number one killer, the kidneys are getting an enormous amount of that blood from the heart. They filter about 4 ounces each minute. They excrete waste, urea and toxins. They retain water, salt, and electrolytes, and they regulate a number of hormones and the homeostasis of many things in the body, right? So the fundamental thing is, how do you define kidney disease when you start with treated ESRD? And by law, the U.S. government decided in the 1970s to treat people with needing dialysis to save their life. This was basically 1972, and the U.S. Congress thought that national health care was around the corner. And these people were dying, so you should pass a law to take care of them, because soon you'll take care of everybody else. Right? Obviously, it wasn't an exact prediction of the future, and it seemed like, oh, it was a small group, so not a big deal to add them to Medicare. Right? It turns out that meant that then the people on dialysis or transplantation were being tracked because the government was paying for their care. So it created an epidemiology of the numerator, treated ESRD, which has a lot of problems because it's not the disease, it's actually the treatment of the disease. So if you're increasing, much of that could just be increased treatment, which is a good thing, or it could be increased disease, which is a bad thing, right? And then soon enough, by 1978, this became 42,000 people, and by the 2000s, this became 680,000 people. So as a rare disease, it's no longer that rare, and in many ways, it's really the tip of the iceberg. So when I was entering this and trying to work with Andy Levy around 2000 on the classification of chronic kidney disease, really, I think the National Kidney Foundation and some of the people who called for this were thinking, we know about dialysis, tell us about pre-dialysis. So they were thinking, oh, you know, there's a condition, 500,000 people, give us another 500,000 or something. We'll work on that and maybe we'll do prevention, right? I think Andy decided that we really needed a fundamental, you know, coherent way to look at this. And he thought that the axis to look at that was decreasing filtration of the kidneys. The kidneys filter. Normal is about 120 milliliters per minute, or 4 ounces per minute, right? And then people with a GFR of less than 15 often need to go on dialysis, so that he decided we would call kidney failure. In medical school, I learned that basically not much happens until you get to 15. It's amazing. You can get by without 90% of your kidney function. Well, the question is, getting by and being alive is not the same as nothing happens to you and risk doesn't increase. So we were trying to ask the question of, when does risk start increasing? When should we define disease? What should we call it? What's the epidemiology of it? And that was sort of the mandate for this group in 2000, which then wrote the guidelines by 2002. So we were sort of fortunate that there was a fundamental question, and there was actually enough data to potentially put this together. And Andy had published on estimating GFR from large populations, so I was able to then connect their thoughts and the structure to the US surveys of large population-based samples to know what cutoffs might mean. What we ended up deciding is to call stage 4 chronic kidney disease a GFR of 15 to 29, sort of double the level of the 15, and there was another 700,000 people. And that was kind of what we sort of thought might be interesting. But then we said, well, how many people have abnormally reduced kidney function? Turns out if you go 2 standard deviations below the normal of 120, you get to about 90. So for a while, there was a Saturday morning phone call where Andy was like, well, maybe we should call it 90. And then I did the math, and it turned out the majority of people would have, at an older age, there was 55 million people with kidney disease. And when I looked at abnormalities, we couldn't detect enough abnormalities, so to me it didn't make sense. So we tried to look at risk relationships and see where the curves shifted for risk. So then we based things on the prevalence of abnormalities, and we said you had to have low enough kidney function and an increased level of abnormalities concurrently. And it turns out that happened at about half of normal kidney function, or 59, right? So we said if you had lost half or more of your kidney function, and you're at a higher risk of anemia, a higher risk of mineral abnormalities, a higher risk of a number of things that we then accumulated into this guideline, then we would call you chronic kidney disease. And that was 15 million people, right? So that was sort of unexpected, and we had to decide, are we going to back down from this, or do we believe it's true, right? And we thought it was true based on all the associations we were observing, so that's where we defined chronic kidney disease. In addition to that, people with more intact filtration, it was clear that having albumin as a marker of damage in your kidney, protein in your urine, was a marker of damage as well, and people with diabetic kidney disease, that was a cause for the progression of their kidney disease, so that was an opportunity for prevention. So it made sense to declare stages 1 and 2 of kidney disease with normal filtration, but an elevated level of kidney markers, and that was another 10 million or so people. So all in all, we came up with about 26 million people, or about 10% of adults, having some stage of chronic kidney disease. And the key was to distinguish that that was not pre-dialysis. It's not that all of these people would go to dialysis. But what was important is they had clear abnormalities in their kidney function that then resulted in adverse events which would potentially be treatable with consequences that would be preventable, right? So we decided to define that as a group of interest and a group that had an abnormality that had consequences, and therefore that was the group to look at. So that was kind of a paradigm shift, and I think that's why it sort of filled a gap in a way that then had to be defended and had consequences over time. What did we have to overcome in making this definition? The hurdles were we needed to get a large nationally representative sample, over 15,000 people, to know what the prevalence was in the whole US of kidney disease. It turns out that was easy. The National Center for Health Statistics was doing these studies, and for $20 you could get the data set, right? So this is where technology became a good thing, because you used to have to work on mainframes. I was invited to a work group that did the first paper using a mainframe, and then I realized, oh, they'll give you the data for basically free, and you can use it on Stata software that was available on our PCs. So then I working on that with a master's student, and we got to do like 100 times more analyses than they were able to do before, and I think we're able to get much more nimble insights into what was going on. Then we had to convert serum creatinine to estimated GFR. And there had been a number of formulas over decades, but conveniently Andrew Levy was involved in designing the most carefully developed equation at that point from the MDRD study. But as soon as I tried to apply it to the N. Haynes study, I realized this didn't work at all. It didn't make sense. The estimates of the normal kidney function were in the wrong place. So then I dug into literature, and it turns out creatinine had been measured for almost 100 years, but its precision was amazing, a coefficient of variation of 2 or 3%. But the bias in the measurements between labs was one of the worst of any measurement in clinical chemistry. It turned out that as a result of the initial measurements in creatinine were 20 to 30% too high, but people got used to those measurements. People said that that was convenient because when you were calculating clearance in the urine, it canceled with another biological problem. So doctors liked the numbers being wrong. Then they came up with enzymatic methods that were correct, but nobody wanted to use them because they were different and they didn't work in that convenient cancellation. So then the industry said, fine, we'll take those true numbers, we'll multiply them up, and we'll get them around where they were the other ones. But there was nothing to calibrate to. So then it was just a very poorly calibrated field, and we wanted to now look at near the normal range. If you just want to look at dialysis, it doesn't matter that much. But if you want to get closer to the curve and look at moderate abnormalities, you need to get the calibration right. So it turned out this boring thing was important, so we dug into it, did some fundamental work, and then published on calibration, and then used the equation using a calibrated method. And again, that was not complicated, but fundamental to being able to do it in a way that would be consistent and replicated and stand the test of time. And then we needed a good equation, and that was published in 1999. So all the right ingredients were there in 2000 when we were doing the work group, and then we did the work by 2002. Then the other thing that became an implication that I didn't fully realize the true impact is that once you define kidney disease to affect 10% of the population, and you have 20 million people where nephrologists are busy taking care of 500,000 people, then kidney disease care cannot be the focus of nephrologists only. It means that people really have to work in a multidisciplinary way and look at kidney disease in its earlier stages as a part of general care, cardiology care, endocrinology care, diabetes care, surgical care, etc. And some of that was realized, but until the equations were in place, it was not fully clear. So that, I think, became somewhat transformative, right? And in some sense, we found out over years that specialists, the nephrologists, loved the idea that their disease was important. So going from 500,000 people on dialysis to pre-dialysis and double, triple, that was fine. But when they deeply thought about 20 million people and the fact that it was no longer their disease, a good number of people really didn't like this and started saying, well, maybe it's not kidney disease, maybe we should just change the definition, right? So we had to deal with that over time. So this is an editorial published by Kai-Uwe Eckhart and a group of us in Lancet, later saying, evolving importance of kidney disease from subspecialty to global health burden. Then it turns out once you define it in the US, you want to look at other countries, and it turned out kidney disease is just as common in other countries as in the US, and with the increasing burden of chronic disease globally, hypertension, diabetes, poor control, you're getting kidney disease increasing globally, and even the dialysis epidemic increasing, even though the numbers are often not as dramatic because treated ESRD is not as big because there isn't treatment. But you're getting lots of people with end-stage renal disease. And at this point, we have more people on dialysis in Asia than North America, right? But half the people who need dialysis in Asia are not getting dialysis. And in Africa, it's even worse. So this was an early model that we realized that you really needed a co-management of patients with CKD moving from primary care to nephrology specialty care. Gary Agnoyan, when we were doing this, insisted on the K and the D, right? I was young and naive, and I was like, what does it matter, right? But he thought we should use kidney rather than renal so people would understand what we're saying, and D because people understand disease, right? And chronic made sense because it was a chronic disease. But it turned out those words actually matter, right? So that was a lot of the translation. This is a cartoon in terms of words matter. I want a potion that will add 40 years to my life, right? So the great scientists here, try this. Right? Let me rephrase that, adding 40 years to my life, right? So then there was the question of if we were going to have estimated GFR and we were going to use the new equation, it turns out the previous equation was beautiful. Cockcroft-Gault developed it in Baltimore here at Hopkins, and there were nice round numbers that you could divide and remember in your head. The new equation had a log and ugly numbers. So it was clear that it was going to be annoying to calculate, but I was thinking, why should any physicians be calculating anything with a pencil or a calculator? It should be automated reporting in the laboratories. Right? The joke is, in 2002, some of the leading laboratorians were telling me, our LMS people may not be able to do exponents. And I was thinking, what do you mean you can't do exponents? It's a computer. It must be able to do exponents, right? So we sort of insisted, and they automatically reported that in their labs. It turns out from 2003 to 2008, we basically went to increasing number of laboratories, and because the guidelines were clear, we asked the manufacturers to calibrate their creatinine, so we no longer told them we didn't want the truth. We told them we wanted the truth. Tom Hostetter led a national kidney disease education program and a standardization effort that told them exactly what they wanted. So by 2009, all the manufacturers were calibrated, and nearly all the labs were reporting this in an automated manner, right? And so in many ways, I think that's the future. If you can get the computers and the main systems to do the work that help the physicians, I think physicians will adopt it rapidly, right? So you need to get consensus to move forward. And later there's an app to do this, but I think an app is actually moving backwards, right? That's the doctor typing things in. I think things should actually happen on the back end in the medical system. Some of the science that was underlying this is really the idea that measured GFR led to estimated GFR, which was reflecting of that, and then that allowed us to predict outcomes. We formed a consortium, the CKD Epidemiology Consortium, to look at the performance of different markers to estimate GFR, and we've spent decades, and we're still working on novel, better and better methods to estimate GFR. It turns out the initial estimate was pretty good. And then by the time we spent 6 years to develop a better estimate that I'll show you, a lot of the people who were the opposition were like, oh, just use the old one. What do you need this new one for? And we're like, we worked for 6 years to get you a new method. Just because it's close to the old method, you could still improve some. And then looking at the outcomes that relate to GFR, and later we formed the CKD Prognosis Consortium to look at the association with all these outcomes, and it's a wealth of outcomes that are really related. So talking about estimating GFR, this is the CKD Epi collaboration, and the key to that is this group of statisticians and nephrologists that work together to do that. So the idea was to take serum creatinine, which in 1975 Cockcroft-Gault had related to 24-hour urine clearance in 249 men, and basically made a guesstimate for women. And that was used as a dominant equation for like nearly 25 years. So it was easy to beat. In 1999, the MDRD study equation was developed by Andy Levy here and Tom Green. And Tom thought, oh, this is just kind of a nuisance. It may not be that important to paper, but he did a really good job. And Andy told him, it's probably going to be important, right? And I think Andy was right, and it's sort of a running joke between Andy and Tom, who are good friends. And then in 2002, we defined estimated GFR less than 60 to be kidney disease, so it became quite important, and we needed to know whether the MDRD equation, which was now developed on a lot of people, but not a very representative sample of the population, was really valid. So in trying to counter those questions, we then spent a lot of time on global standardization of creatinine, routine reporting of kidney function, and in 2009, an improved equation based on a consortium of 12,000 individuals with measured GFR from 30 studies. And then it was implemented in the largest US labs and more. So to develop the CKD equation, we got a set of studies to develop. We got internal validation in the other 1/3 of the data, external validation in other studies, and then we got the final model. This paper was published in Annals in 2009 with Andy as the lead author and me as the last author. And it turned out the MDRD study had a bias. If you look at estimated GFR and the error between measured minus estimated GFR, Below 60, it was not terrible, and that's where we used it to define kidney disease. Above 60, there was a consistent bias. And it turns out, having worked for 5 years, we could cut that bias in half. So we basically cut the difference from 11 milliliter difference to 4 milliliters difference in the error rate substantially. On the other hand, it wasn't like dramatically different, and in the important range below 60, it was quite similar, which in a way was convenient because it meant that everything we said wasn't wrong, it could just be incrementally improved. So in the estimated GFR for the US population here in 2009, we say this was the MDRD estimate of the distribution of GFR in the population. The CKD Epi estimate is very similar up to here, like 45. It's a little bit lower below 60. And then in the normal range, it's different because it had a lot more normal people. So the influence on the prevalence of kidney disease was to decrease the prevalence of kidney disease just a little bit. And it actually did that more in the younger age. But in the older age, it left the estimate at nearly 50% of people aged 70 plus, having some form in the whole spectrum of kidney disease, right? Not just ESRD. Now that led to a whole controversy. And basically, we needed to figure out what happens if you have kidney disease, because then, is it really a disease? Is it really a problem? Or some people talked about healthy aging. We looked at this model of basically progression of kidney disease along the x-axis, getting these 20 million people, but it was clear that kidney failure was only a minority of the issues. And really a dominant pathway was complications, people with albuminuria or low GFR having strokes, heart attacks, heart failure, and going to death around dialysis. So if you stayed focused only on dialysis, you're only looking at a skewed part of the picture. And then there was sort of some vocal opposition, which later declared themselves to be the loyal opposition. And it turned out that was not unuseful to us. But there was a debate on the epidemic of chronic kidney disease, the challenge. And they said the challenge, an epidemic of chronic kidney disease, fact or fiction, and argued that a cutoff of less than 60 without any correction for age or gender, without any consideration of concomitant albuminuria, was wrong, and a substantial revision of the guidelines from 2002 is overdue when they published this in 2008, and it's really normal decline in EGFR with aging. Our response is that we needed to look at the full range of CKD. If you want pre-dialysis, yes, 60 is not the right number. And it's true that the high numbers are beyond the scope of nephrologists alone. But we needed to refine risk estimates and distinguish optimal from normal. And for me, this is where I learned lessons from Paul Welton. Hypertension is nearly universal if you get old enough. But the SHEP study showed us that if you take somebody old and you treat them, you prevent strokes. So it may be universal, it may be extremely common, but it is abnormal, treatable, and you can reduce risk. Cataracts become increasingly common. And later, when I work with Frank Lin, hearing loss becomes increasingly common. So if we declare all of these to be normal, we would say normal aging means that you get hypertension, stroke, cataracts, blindness, deafness, and kidney disease. Maybe these are actually diseases, and there's a challenge that we need to address. So we need to target the most promising interventions for clinical trials, treatments at the subgroup at higher risk, which would benefit, and it's premature to use a high prevalence or lack of proven treatments to define individuals with reduced kidney function as quote-unquote healthy, right? So then this was a controversy, so we decided to have a controversies conference. And they put Andy on the planning committee, and initially they said, well, you don't need Joe because Andy and Joe are kind of the same thing, so you don't need him. Conveniently, Andy's a good friend, and he said, you got to have Joe. You can't just, I'm not doing this by myself. So he brought me in. And then we had a planning committee, and we decided it should be about data. If it's not about data, we'll just come in arguing, we'll leave arguing, and everybody will believe what they believe. So we decided it should be about data, and it should be about risk, because that's what we can define. So everybody who comes to the meeting should bring their data. And then we decided everybody had to do a common analysis. And then we realized if we wanted to do a common analysis, we had to do the analysis. So then we volunteered to do the analysis for free for everybody on time. Turns out when we did that analysis, we first of all realized we didn't know how to do the analysis, so we had to learn. We taught ourselves. We did the analysis by the meeting. We came to the meeting with 2,000 slides of original analysis for every single one of 30 cohorts, and then a meta-analysis for everything with like over a couple hundred slides, meta-analyses for low-risk studies, high-risk studies, CKD studies, and clinical trials separately. And it turns out there was consensus. So this is the results for the meta-analysis in general population cohorts, and one of these was published in Lancet in 2010, and then the other 3 papers led by colleagues on the planning committee for different outcomes. And basically, if you looked at eGFR on the x-axis, and you put a knot here at 95 as the reference group, if you had no albuminuria, the risk increased, and by 60, the confidence interval for all-cause mortality was no longer crossing 1, it was statistically significant. Interestingly, if you had albuminuria, the risk was higher, but the GFR relationship was miraculously almost parallel. If you had higher level of albuminuria, the risk was even higher and miraculously almost parallel again. And as a numbers person, for me, that's just like beautiful, you know. And then if you looked at cardiovascular disease, there's some differences, but generally similar. If you looked at end-stage renal disease, similar pattern. By 60, the relative risks are higher. And if you go at 15 versus 95 for estimated GFR, the relative risk is as high as 8,000. So this estimated GFR, which had gotten a lot of criticism, is imperfect. But for a very specific outcome, like end-stage renal disease, you have an 8,000-fold gradient, which we said it's worth persisting, and you can use it for staging as we proposed. And clearly, being at an earlier stage is not as bad as being at a later stage, but there could be something to be done. And 60 was significant at every group and at every age, and then we continued to do more of that. It's still taking a while to change things, but the later guidelines that came out supported this, right? So it turns out we presented this. There was actually consensus at the meeting, but then it became clear that we shouldn't kill this collaboration that got created. So we named it the CKD Prognosis Consortium. It's now increased, and every year it's growing. So now we have 80 cohorts and about 10 million participants because big data is a whole movement. We now cover most of the world, but we need to do better in Africa and China. We have annual meetings, and all of these leading scientists contribute. The number of people and cohorts continues to grow. As a consortium, we established certain structural elements, and I think that's what's helped things continue. It needed to be open. It needed to be fair. We rotated first and last authors. We limited our writing groups to the kind that can work. We acknowledge about 200 members of all the different cohorts that have worked for decades to make the data possible in the back of the paper. And we hope that the analysis is rigorous. I've learned from somebody that actually all the details are incredibly important, but that's not what everybody wants to see. So I've learned from the GWAS work and the genetic work, have detailed appendices where you show all your details, and then have clear top messages in the paper itself. And we do lots of analysis to try and inform. And then we had the rule of not subtracting from leading cohorts, because if you want cohorts to contribute to the group, you can't tell them not to publish their own results. So you had to encourage people to scoop you first, and then hope that there would still be something important for you to say. But at least in epidemiology, the definitive word is not the first word. In basic science, it may be a little different, but I still believe to get to a clear understanding takes some work and takes some consensus. So we encourage people to do and test our innovative concepts and tell them a year ahead of time that then we will test it definitively in everybody else. And we work in phases to try and have definitive goals every year. And we take requests. And it turns out some of the highest impact papers were requests by others. That led to the guidelines, which then led to the risk by GFR and albuminuria, as we suggested, and the cutoff of 60 has actually not changed at all since 2002. We then looked at the new equation, the CKD Epi equation. It performs better than the previous MDRD equation. We then looked at additional markers like cystatin, and it turns out we had Mike Schlipak, who did the pioneering work on cystatin, lead that for the consortium, and it turns out cystatin indeed is a better predictor of mortality. These two lines here in red and blue are stronger than the line in black for GFR. So by a GFR of 60, with cystatin, you actually detect more people and a higher risk. But for ESRD, it turns out creatinine was every bit as strong as cystatin. So it matters what you look at. We looked at a global validation of a kidney failure risk equation. If you want to stratify people at risk, Nav Tangri had worked with Andy Levy in 2 Canadian cohorts. We helped him expand that to a global consortium to see how things work. We looked at discrimination and we looked at calibration and found that in Europe, on average, for the same risk factors, the risk of dialysis is much lower than in North American cohorts. So we were sort of extending that work. Morgan Grams, who joined us as a fellow, became a leader and now is a co-PI of the consortium and leads a lot of the work. And she looked at lifetime risk of CKD, ESRD, and that concept has generalized. And then there was the question of how do we help patients? So as an observational epidemiologist, all we do is describe. The hope is that people would take that and then use that to help people, but describing alone needs to do something with it. So we decided to look at how do we accelerate clinical trials, and in collaboration with the FDA, we tried to look at the outcomes for kidney disease. So it turns out doubling of serum creatinine was used as an outcome, and that results in a 31-fold increased risk of dialysis. And we found out that a 30% decrease in GFR was much, much more common. Doubling was present in half of 1% of people. Doubling was present, a 30% decrease was present in 5%, so 10 times more common, and yet a 5-fold increased risk seen across many cohorts, consistently in low GFR and high GFR. And it's really this consistency that's driven the ability to make conclusions that were then accepted by regulators. Finally, the last couple of slides, right? In some sense, while I do all this kidney disease, as a vascular epidemiologist, the heart is still the leading cause of death. So our ERIC cohort is still doing a lot of work on cardiovascular disease in the heart. We expanded to the brain, and now we're doing a lot of papers on the brain. And then there was a twist recently. 5 years ago, I met Frank Lin. He gave one of these talks and showed that over the age of 70, hearing loss is present in the majority of people. And I kind of knew that, but I wasn't fully attuned. And then he showed that hearing loss was related to rapid cognitive decline and increased risk of dementia. Right? And it became obvious that whether you could correct that with hearing aids, not just the hearing loss, but the risk of dementia was sort of fundamental. So the same day we decided to meet upstairs after the seminar, we decided to help each other, connect the ERIC cohort and the staff we had with his lovely ideas. We started a pilot a month later. We installed a hearing booth. We got that data. We used that data to do a pilot of a trial. And then we got in 2017 funding for a large randomized trial to test exactly this concept. And in many ways, it's really Frank's ideas, but I've had the pleasure of riding along and making it possible. So if you build tools in many disciplines, taking those tools and moving them across, I think can be transformative. At least we'll have an answer, and we should have one by 2022. This is a typo. But we're halfway past randomizing the people. So we have over 400 people already randomized to this trial. So I talked about epidemiology being at the interface, and I hope I showed you that it can be a basic science for clinical research and informing the health of the public. I like focusing on answerable, important questions. And as to the future, I think we'll have to deal with big data and a number of other issues. But I think if we embrace these emerging technologies, I think we can cleverly and collaboratively make progress on big problems. So by way of acknowledgement, right, this is really team science with many big teams of wonderful people. I've been very fortunate to meet them here at Hopkins and elsewhere. And these are some of the key teams in the CKD Epi collaboration, the CKD Prognosis Consortium, pictured here, the Coordinating Center group, the staff in Hagerstown to collect the data, the cardiovascular epidemiologist, and an early picture with Mike Clagg and Neil Poe, who were my early mentors. Thank you. Questions?
I wondered whether you had guidelines separately for males and females, or is it just one common?
Yeah, so we decided to have one common cutoff. In nuances, there actually is a difference, but we decided at some point the trade-off between complexity and the impact to stick to the simpler guidelines. Yeah, I think we may not have much time.